In this video, we'll be seeing how to look for the combinations gene-by-gene modulator that have resulted from a completed analysis. First, we need to click on the green button from the Actions column to access the Results panel. A new tab is then opened by the system next to the All Analysis tab. If we wish to return to the previous panel, we'll just click on All Analysis again. Keep in mind that several tabs may be open at the same time. If we wish to close the tab, we can do so just by clicking on the X. Let's return to the Example tab. The first column on the main panel shows information regarding the expression modulator. In this case, it's a microRNA, since the analysis involved this molecule and mRNA. In the following columns, we'll find the name of the gene and data regarding this last molecule, including the chromosome, the start and ending positions, the type of gene, in this case it encodes a protein but it could be a pseudogene, and a column with a long description of the gene. We may hover with the mouse over the cell for more detailed information. Further to the right, we'll find the columns that refer to the correlation analysis, such as the correlation test, in this case it was a Pearson correlation, then the p-value and the adjusted p-value. In this case, for example, the Benjamin-Hochberg technique was applied. On the top, we'll find how many combinations resulted with the applied filters out of the total number of combinations for the analysis. There's a search field for gene or modulator, a filter for the correlation coefficient threshold, and a select for positive, negative, or both types of correlation. For microRNA and methylation analysis, correlations that will be shown by default are negative ones, whereas for CNA, it's positive correlations. There's a button that allows for a simplified view of p-values that shows if the value is below statistical significance level. The yellow button on the right resets all applied filters. Keep in mind as well that the panel allows users to order the results by any column. By default, it's a descending order for absolute values in the correlation column. If we click again on the header, the order is inverted, and a third click will eliminate this order sorting. When we apply sorting by descending or ascending order, this will be shown on top of the panel. We may combine more than one sorting order, in this example a descending order for microRNA and an ascending order for mRNA at the same time. If we wish to clear this sorting, we'll just click on this red button. Both filters in the top and sorting order for the columns may be deleted by this red button on top for a full reset of the view of results to the default descending correlation order. If we wish to download the data with the applied filters and sorting that we see on screen, this blue download button provides such functionality. For example, if we apply this filter, we'll be downloading a file with just this single tuple. Let's clear the view again. Finally, the Actions column deploys a new panel with further information on the combination of interest. First, we'll see a panel with statistical data, such as the average, standard deviation, and results for the Shapiro normality test, both for the gene or mRNA and microRNA. Areas may be deleted using this first button, or both graphs may be integrated in a single unified one. When we go downwards, a box plot will appear, revealing more information on the median and the outliers. These values were obtained by the method of median absolute deviation. Further down, we'll find the number of evaluated samples and the results for a test of Bruch-Pagan and Goldfield count that indicate heteroscedasticity and homoscedasticity, as well as linearity and the monotonicity test. The latter is the Spearman rank test. There are help buttons to get more information regarding each statistical test. We'll also find a correlation graph with a linear regression that adjusts the expression of the gene and the modulator. The functionality of this panel consists of being able to color the samples in the graph using the input from above, after having uploaded to the analysis the corresponding clinical data. This process, as well as the survival analysis, which also depends on clinical data, will be explained in detail in another video. Other tabs in this panel show interactions between the gene and the modulator, disorders and drugs. This tab shows the result for the interaction between mRNA and microRNA, including the score for the interaction inferred by MIRDIP, a resource that has registered over 152 million predicted interactions for such molecule tuples. On the right, we'll find the sources MIRDIP has consulted to calculate the score, and further to the right, scientific publications that refer to the microRNA and gene are listed. The next tab shows known interactions between the microRNA and any other gene, not only the one that has been correlated in the current analysis. This may add relevant information for interpretation of the results or provide us with new hypotheses. Here we'll see the genes it is known to interact with, the sources of the data, the score for the interaction inferred by MIRDIP, and the class of the assigned score. The results may be sorted by ascending or descending order, and in the last column the scientific publications on the interaction will be shown. The last tabs show interactions between the microRNA and medical conditions and drugs. The aliases for the microRNA are shown, as well as the sequence. If we click on the alias, Mirbase will open and show us more information on this molecule. These last tabs are based on information retrieved by Modulector, an open source application developed by our team that integrates multiple data sources for microRNA and allows us to show information with high performance, agility and consistency. The last 4 tabs on the right are only available for experiments that include microRNA and will not be shown for other modulators such as CNA or methylation. The Survival Analysis tab will be explained in other videos. In the special cases that include CNA analysis, if such data is categorical, the system will detect this automatically and show results as a box plot instead of a correlation graph with an adjustment line. Here, if we hover the mouse over the element, relevant information will appear. Violin plots can also be shown or hidden if we wish to.